and then also your mentor.
Awesome, thank you. So, hi, my name is Eva. I'm from California, and it's in Boston College. Thank you, Nicole, for organizing the symposium. My mentors are Dr. Aaron Bredeweg and Scott Baker. You may have heard the quote, "A loaf of bread, a jug of wine, and thou." Well, everyone except thou is provided by our friend, the yeast. My plan today is to talk about how yeast deal with stress. I'll begin with the background, our experimental design, results, next steps, but we'll be able to track the progress at the top of the screen. As I'm going through, feel free to write any questions down, and I'll do my best to answer them at the end. So first, let's talk about essential genes. These are like your muscles and bones. They serve fundamental functions, and you don't change them unless you absolutely have to. In fungal species, they constitute a fifth of the genome, or about 22% in our yeast, Yersinia lipolytica. Now, essential genes are important because they target critical pathways. This is valuable for three reasons. First, in Yersinia, an oleaginous yeast, investigating essential genes is key to biological processes for hydrocarbon-based chemicals, biofuels, the fuel of the future. Second, they reveal ways to fight fungal pathogens. And third, and of special interest today, they behave differently in genetic studies. We'll investigate this further by using a common genetic experiment, transformation. So this is Yersinia. That's about 5 micrometers in diameter on the left, about 15 microns on the right. When it gets stressed out, it samples the environment and looks for genes, like how people seek others when they're feeling anxious. In our transformation, we're going to stress it out on purpose, so it takes up the DNA we give it, allowing exogenous DNA to be, or a DNA outside of the cell, to be incorporated into its genome. However, the principles of transformation experiments are not well understood, a black box per se, and fungi are more challenged to transform because of traits like a thickened cell wall. For example, even if the DNA got into the cell, sometimes it goes to the wrong place in the genome, resulting in ectopic or abnormal integration. Cells with ectopic integration will still grow, resulting in false positives. This is because of non-homologous end joining, a DNA repair mechanism that blindly inserts DNA without regard to its sequence, like cleaning a mess blindfolded. As such, the yeast we use has this mechanism defective, which should limit the false positives. We need to investigate conditions for improving transformation techniques and advancing genetic studies on essential genes. This study addresses this by posing the following questions. Does DNA concentration, 1, or using single-stranded or double-stranded carrier DNA, 2, increase colony yield and transformation efficiency? Colony yield is the number of colonies that grow following our transformation experiment, and transformation efficiency is the measure of successful integration of the exogenous DNA, or testing the number of false positives from ectopic integration I mentioned before. To answer these questions, first we tested the effect of varying concentrations of DNA for 3 essential and 9 non-essential genes on colony yield. Then we tested the effect of single-stranded or double-stranded carrier DNA on colony yield and transformation efficiency of the essential gene Fas2 and non-essential gene ACA2. To accomplish this, we used split marker PCR to insert a GFP, that's green fluorescent protein construct between 2 gene flanks, essentially sticking a flashlight on a protein, and amplified or made copies of segments of the DNA piece. However, this new piece is still outside the cell. So we used transformation techniques by stressing out the cell so it would seek DNA, and then added the GFP construct to its environment. The gene flanks guide the PCR product by homology so that it integrates into the genome at the correct location. Now, both conditions took place in the transformation reaction. The first in which we varied the DNA concentration or the amount of this product, and the second in which we used either single-stranded or double-stranded carrier DNA. To measure colony yield, we used a CFU AI. For the carrier DNA, we tested both colony yield and transformation efficiency, or as I said, the measure of successful integration of exogenous DNA, or false positives, by screening isolates with PCR. And from our experiment, we were able to consider a few observations. First, increasing DNA concentration increased the average colony yield of the 12 genes. This was expected. Adding a higher concentration of donor DNA increases the amount available for uptake by the recipient cell. So the greater amount of DNA in the recipient cell would enhance the chance of integration, which should increase colony yield. Second, essential genes, the 9 non-essential genes, had a higher colony yield than the 3 essential genes. The average colony yield for essential genes was 3.23 colony-forming units, CFU, per femtomoles, a measure of DNA, and for non-essential genes was 5.8 colony-forming units per femtomoles. This is because adding a GFP, green fluorescent protein molecule, to a protein is generally understood to be a hindrance to a protein's function and decreases its efficiency. So modifying a protein that's essential to a cell will more significantly impact fitness and select against survival. The low colony yield is expected for essential genes because transformants would be less likely to survive. However, modifying a protein that's non-essential will not select against survival as strongly, making transformants more likely to survive. When testing single carrier DNA, we found that single-stranded carrier DNA significantly increased the colony yield of ACA2 and FAST2. It doubled the average colony yield for the essential gene FAST2 and tripled the average colony yield for the non-essential gene ACA2. However, colony yield does not correlate to transformation efficiency. Remember, that's whether or not the DNA correctly integrated into the genome. Despite the 2-fold and 3-fold increases in colony yield, the transformation efficiency did not significantly increase. For the non-essential gene, ACA2, the unboiled, the single-stranded and double-stranded reactions had 5 and 6 successful integrations of the 8 isolates tested. For the essential gene FAST2, there was no successful integration of DNA across the 8 isolates tested, regardless of if we use single-stranded or double-stranded carrier DNA, which suggests that all transformants from the essential gene FAST2 resulted in ectopic, or the abnormal integration. Even though it appears that single-stranded DNA increases the transformation of ACA2, it had no effect on the transformation efficiency of the essential gene FAST2. So in this paper, we investigated the effect of DNA concentration and the state of carrier DNA on 2 measures of DNA uptake: colony yield and transformation efficiency. Our results suggest that increasing DNA concentration and using single-stranded carrier DNA increases colony yield more for non-essential genes than essential genes. Further, despite doubling the colony yield, single-stranded carrier DNA did not impact the transformation efficiency for the essential gene FAST2. These findings offer new observations on differences of DNA uptake and integration between essential and non-essential genes, which will guide conditions for future experiments on genes with relevance in both the biofuel and medical industry. Further, these results are informing our protocols for our next project. We'll be able to use these observations when targeting ALAS, a non-essential gene, in urea with transformation methods. ALAS is involved in heme biosynthesis, a key molecule in iron homeostasis. And by using transformation methods to produce multiple urea strains targeting ALAS, we'll be able to make a few observations. First, we can compare the localization and expression levels using GFP by microscopy. And second, we can test the phenotype of an ALAS mutant when challenged with high oxidative stress, in our case, hydrogen peroxide. So I'd like to thank the Pacific Northwest National Lab for providing the resources for me to research on their campus, my phenomenal mentors, Dr. Aaron Bredeweg and Dr. Scott Baker, Nicole for coordinating the SULI program, and the Office of Workforce Development for Teachers and Scientists for funding the SULI program so that I was able to complete this research. Thank you for listening, and I'll go ahead and prepare for any questions that you might have.
Thank you. Thank you, Eva. That was wonderful. We have a question from Laura Lowry. Great talk. I have two questions for you. First, big picture question. Sorry, I'm not that familiar with oleaginous yeast. Can you comment on the benefits of studying transformation efficiency in this particular type of yeast as opposed to more traditional yeast model systems?
Yeah, so I think there's two parts to that question. The first is, why is Yaria as an oleaginous yeast important for scientific research? So when creating biofuels, Yaria has been known to both be able to break down oils in the environment and also produce lipids that can contribute to the biofuel biodiesel. So scientists have been investigating key metabolic processes in Yaria so that we're able to improve using Yaria for biofuel advancement. And the second question is, in order for scientists to understand those key metabolic processes, we need to have an understanding of how to manipulate the genes in Yaria using various genetic methods, with the most common being the transformation mechanism that I described.
Okay, thank you. Question from Macy Schmidt. What was your favorite part of conducting this research?
My favorite part of conducting this research, the first is that I learned a lot. I'm really grateful because my mentors are phenomenal teachers, so it felt like I was learning 3 or 4 classes a day every time I went into lab. And my second favorite part was having a broader understanding of the various applications of fungi in being able to complete an experiment and kind of work with a living organism. They don't behave the way that you want them to sometimes. So it's really interesting.
Okay, a question from Antoine Davidkins. Says, hello, Eva. Why do you think the non-essential genes were more successful with integration versus the essential genes?
Yeah, that's a great question. We saw that essential genes had a lower colony yield than non-essential genes, which plays directly into, of the amount of colonies that formed, which ones were successful or not. I think my biggest understanding is that when you add that GFP molecule to the end of an essential gene, and then that gene is transcribed into a protein, then the protein has to continue its regular function but essentially having like a backpack of rocks on its back. It has to carry this GFP molecule with it when it's doing the essential function. So one of the reasons that it had such lower transformation efficiency is that when all the colonies that had to carry that backpack didn't survive or didn't show up on our culture plates because the GFP molecule hindered its function so much that it impacted its survival. While for non-essential genes, carrying that backpack doesn't have to impact the overall well-being of the cell as much, and it can still grow on the culture plate and we